Dear students, welcome to the basic genetics course, and we are discussing currently the gene mapping in prokaryotes. In this unit 6, we are talking about various ways through which we can map the genes in the prokaryotes, and especially we are taking the example of bacteria. So in this class, I will be talking to you about the conjugation and sexduction. These two methods, which are very closely connected with some minor differences that I will highlight to help you understand how exactly these methods are used for the gene mapping in bacteria, and the example will be mostly E. coli or Bacillus subtilis, these kind of bacteria. Okay, so in the last class we talked about the F factors and HFR. They were plasmids that either stay outside the bacterial genome as an independent entity, or they have the capability to get integrated, and when they get integrated we call them as HFR. So F plasmid, as you can see, they carry the genes that are required for the formation of this kind of sex pilus, and they are also required for the transfer of bacterial to transfer the DNA from one donor bacteria to the recipient bacteria. Right. So these are the F plasmid, and I also told you that when a F plus donor conjugates with the F minus recipient, the F minus become F plus, and I did inform you that when a HFR donor conjugate with the F minus recipient, the recipient always remain F minus, and I in very detail I did also mention this happens because the complete transfer, because when the F factor integrates with the bacterial genome, then what happens is that the genome is larger compared to the F factor alone. So if it is only the F factor that has to be transferred, it is easy for it to get transferred and F minus cell become F plus, but when it get integrated become HFR, then it is almost impossible to transfer itself along with the whole genome, and that is when the F minus cell never becomes a F plus cell. And I also did tell you that these HFR strains, they are used in the mapping of the bacterial genome. So these HFR strains, they can inserted at different locations in the bacterial genome, right? And using this quality, I'll show you in detail, this quality of these HFR strains, that the F factor can get integrated into the bacterial genome at different places, making these HFR strains. You can use different HFR strains to map the bacterial genome. And the other important point that I mentioned, and I'll discuss that in detail today, that these transfers are usually non-random, right? The genes transfer was shown to vary from one HFR to another HFR, but it is always directional, right? So what does that mean? We'll come to it. So what kind of experiments people did when they talk about the gene mapping in bacteria? So these called the interrupted mating experiment. Means mating, you already understood that you have a bacterial cell and then it connects with the recipient, another recipient cell, and then it transfers DNA to another. And this is happening because of a tube which we call as sex pilus, right? So what if you disrupt this tube, right? So if you disrupt this while the process is going on, then what would happen? This bacteria will go other side, that bacteria will go other side, and that way this transfer will be interrupted because you have broken the contact, the communication between the two bacteria, donor and the recipient. So that is called the interruption. And in an experiment, how usually we do this kind of interruption, like when these people have done this kind of mapping, so they allowed the bacteria to grow, right, and then both the strains came into the contact, and what they did is they took this solution and then this solution was transferred into a blender, right, and then in a blender when you give a high speed, then the bacterial cells get vigorously shaken and then the connections between the two cells are broken, right. So that is called, remember that is called interrupted mating, and you can do this kind of mating, you can interrupt the mating at different time points. You can interrupt it after 5 minutes, after 10 minutes, after 15 and so on. So usually it takes about 100 minutes to transfer the entire E. coli genome. So between this duration, whenever you will interrupt, what would happen? As much time you allow the bacteria to remain in the conjugation phase or connected through the sex phylus, the DNA kept on transferring from one cell to another. And so more the time you allow, more the DNA will be transferred. And when you break, this transfer will be interrupted. So chromosomal transfer always begins within the integrated F factor and proceeds in a continuous direction. See, this point is important, and that is why it is being written multiple times, right? And what happens is that when the fragment of the DNA is transferred from one bacteria to another, right, it has to be integrated. A linear DNA cannot survive in bacteria for too long. So to become the part of bacterial genome, it has to undergo the recombination with the chromosomal DNA. So the experimental setup for interrupted mating experiment, just for a quick look, are you mix HFR with F minus cell, allow conjugation to begin. I just mentioned you interrupt the conjugation in using a blender or anything which has a high speed and vigorous shaking, and so you stop the conjugation, and then whatever the bacteria you have, you plate them on selective media plates. See, I already discussed this concept of different type of media, where the minimum media, or complete media, or media supplemented with different kind of amino acids. So this is what they are saying. So you can always plate the bacteria on different types of media plates to see how much, on what kind of bacterial media plates supplemented with specific nutrients these bacteria are actually growing. So now let's look at an example, right? How we understand this ordered transfer of genes by using a conjugation experiment. So pay attention to this HFR strain. What you see that this HFR strain has threonine, leucine, and it has a resistant gene for azithromycin. It is TON sensitive, which is required for some metabolic processes. It is LAC positive and it is GAL positive. Whereas the recipient, see, the recipient is F minus. It is threonine negative, leucine negative, and it is sensitive to azithromycin. It is resistant, just opposite to what you see in the HFR strain. It is resistant to TON. It is LAC minus and GAL minus, right? So now when you have two bacteria with different sets of alleles for same genes, we can see what get transferred and what was still in a process of transferring. So what did they do? Exactly the way I showed you before in the previous flow chart, where I was showing the experimental setup, that you allow the bacteria to combine, right? These two bacteria was connected with the sex phylus and conjugating, and these conjugations were interrupted at 10 minutes, 15 minutes, 20 minutes and 30 minutes. So now how you will know? See, this is just here, this order is just a written order. It doesn't mean that these genes are arranged in this orientation. That is what we need to understand, and that is what we are going to figure out by this experiment. So you see, when the mating was interrupted at 10 minutes, there is only one gene. These genes are color coded. So you can see this blue is for AZI. These cells, bacterial cells, become positive from sensitive to resistant for AZI. No other gene was visible. After 15 minutes, there are even more bacteria that shows the AZI, azithromycin resistant, compared to the azithromycin resistance, and they also have this another gene showing up, which is TON, right? So they become TON sensitive for some of the bacterial type. After 20 minutes, they have another gene added, which is LAC. After 30 minutes, there is one more gene added, which is GAL. So you can see that at 30 minutes, this bacteria has 4 genes, although the maximum that it is showing for AZI because it got the maximum time to transfer. Now most of the bacteria are AZI resistant, and now they also start showing the gal positive phenotype, right? So with the time you start seeing that genes are showing up in a linear order, right? So that is why this time mapping is very important, and initially this mapping method was used for the mapping of E. coli genome. So now coming to the point where I said that we can use multiple HFR strains for the mapping of E. coli genome, and I also said that HFR, this F factor in the bacterial genome, can integrate at multiple different places, right? It's not just one place where it can get integrated, but it can be integrated many places in a genome, right? And so what does it mean? So if you pay attention to this circular map here, then you see I have shown these genes that are arranged on this map, right? So first of all, initially it was not known that bacterial genome is circular. You will see how it was discovered that this genome is actually circular, right? But for our easy convenience, I have just put it the way the bacterial genome actually is. So you can see in this particular genome, the F factor got integrated at this particular location. So in this HFR strain, the F factor is integrated here. Here means where? Between these two genes, leucine and azithromycin, right? And there is this arrow. Pay attention to the direction of this arrow because it is going to help you understand which gene is going to be transferred first and which is going to be transferred next. That is something confusing because just by looking at it, you don't know if you think this gene is going to go first or this gene going to go first, right? So this is something that you need to remember, that the direction of the arrow, as you can see here, tells you that this is the gene just in front of the gene where the arrow is being placed, that gene goes first. Then, so this is first, this is second, this is third, this is fourth, this is fifth, this is sixth, and finally this is the seventh, the last. This one is the last and this one is the first, right? So this is something very important. And if you see the other bacterial genome, other strain where the F factor is inserted here, right? Where this F factor is now inserted between gal and lac gene, right? And now you understand gal will go first, histidine will go second, threonine third, leucine fourth, AZI fifth, proline sixth, right? So that is how this order, and lac will be seventh, right? So last, to remember this point, remember wherever the arrow is pointing to is the gene that is going to transfer at the last, right? So this is something you need to remember. So now I said that the direction of transfer is always directional, right? So you can see after leucine, threonine will be transferred, then histidine, then gal. That has a linear order to follow. So now using this order, these two different orders that I have written here, see the same genes, just pay attention, they are exactly the same genes in these two different strains. But the order of gene transfer is different depending on where HFR is placed. And using this, I will show you how we can understand the orientation or the position of the gene on the chromosome. So you see in this example, when an interrupted mating technique was used with 5 different strains of HFR bacteria, you can see these are different 5 strains, the following order of gene entry was recorded, right? These are different orders and different HFR strains. So they want you to draw a map of bacterial chromosome, and you want to tell it, does it say that this genome is circular? So you see, if you put TCHRO for the first one, for the second, just put this exactly the same way that is matching, right? So then you have ROMB, H is also here, HROMB. For 3 is MORHC, and for 4th it is MBR. B, A, K, and then see T. T is, you see, T, where is the T? T is here. T is here. And then in the fifth one, if you see, it is C, T. So if I would say C, T, K, A, B, right? So that shows, because it is getting connected like that, that shows the bacterial DNA is actually circular. And now there is another example. Many times these small examples come in the examination for you to solve. So it is a good idea to have a look at least a few numericals to understand them. So in a HFR and F minus cross, valine enters at the first place. So let's say valine is the first. But the order of the other markers are unknown. So if the HFR is the wild type and F minus is the auxotrophic, for each marker in question, means it can only grow if it has that particular gene transferred. What will be the order of the markers in the cross when valine recombinants are selected, which I already have written? And then you have trip, which are 25%. You have his, which is 15%. You have threonine, which is 81%. And you have thymine, which is 2%. Right? So the least is this. So this must be the most distant one. The closest is this. It's valine and then threonine. Then comes the TRP and then comes the thymine. So 1, 2, 3 and 4 and finally this is the fifth. Right? So just by looking at the percentage of the recombinants that you are getting, the maximum number of recombinant you will get for those genes that are close, right? The least recombination will be those that are further away. So just summing up, strength of conjugation mapping is that relatively fast, inexpensive, it gives you a good resolution. In 1 minute you can map 40 to 100 kb, and it revealed early understanding of bacterial chromosome. I told you that it is used for the sequencing of bacteria, but there are always some limitation to every technique because not all the cells complete the transfer. That is why it is a bit cumbersome, and the direction is always one directional, and recombination has to occur. As I said, whatever the DNA is getting transferred in the bacterial genome, it has to be recombined with the parental chromosome to show the phenotype, because linear DNA, if it is there, then it will be eventually degraded. And integration side of HFR may affect the observed order, right? So now we are coming to the next continuation to the conjugation process, which is the sexduction. So the same thing, you see, we have F factor, we have HFR, and now we are coming to sexduction, which we denote as F prime, right? So what happens? You already learned that the F plus strain become a HFR when the F factor get integrated into the bacterial genome, right? And what happens here? Sometimes this integrated F factor also tries to come out from the bacterial genome. So if it is a precise excision, fine, you will have again the bacterial genome and you have the F factor separate. But sometimes what happens, because of the imprecise excision, some of the bacterial gene, like in this example LEC, come along with the F factor. So now the F factor is not only having its own gene, which it definitely has, but it also has some genes that are part of bacterial genome. And that is why we are not calling it as F plus, because F plus has a specific meaning. We are calling it as F prime, right? So this is the F prime for the sexduction. So sexduction process, F donor transfer, F dash donor transfer entire F dash plasmid to the F recipient cell. So what does it mean? So if this thing is a part of a bacterial cell, right, same way as the F plus, when it combines with F minus, the F plus F minus become F plus. Same way, this small F prime plasmid will also be when transferred in a F minus cell will make it a F prime cell, because it can completely transfer, unlike the HFR strain. So both the cells become F prime. Any genes on the F plasmid, including those from the bacterial chromosome, may be transferred to the F minus cell. But see, there is one very interesting part, that bacteria already have its chromosomal DNA, right? It has its genomic DNA, and it has this F prime factor which carries one of the bacterial gene. And because of that, you see, for this particular gene, the bacteria is diploid, right? Because usually bacteria are haploid, they only have one copy of the gene. But because of this presence of extra copy, the bacteria become diploid. And there is a specific term used for these partial diploid. Why partial? Because only few genes that are present on this F prime will be duplicated, right? Will be in diploid form. They are also known as merozygotes. So what are merozygotes and how they are formed? This has been shown in this diagram. You can see there is a HFR cell where the F factor is integrated into the bacterial genome, and because of the imprecise excision, you have this F prime cell where a small part of the bacterial genome is integrated and become the part of F factor, right? So these A and B from the bacterial genome, they get integrated into the F factor, and then when this F factor come into the contact of F minus cell, and they make these 6 pilus and start transferring their DNA. Remember, I told you the transfer point is ODT. So when they start transferring their DNA, what happens? This F factor along with the bacterial gene get transferred into the another F minus bacterial cell. And then what happens? This F minus bacterial cell, which become F prime, it has allele of this. So you can see that it has, it was not B. So you can see A, it has capital A, and the genome has small a. The plasmid have capital E, and the genome has a small e. So for A and E, this bacteria now become the partial diploid. Right, so remember this word merozygote, and also why they are called as partial diploids. So advantages of sexduction: they are efficient gene transfer method, right? It creates a stable diploid for the study, right? Diploids, because see, in a haploid situation, you may not be able to understand which is dominant, which is recessive, because in a haploid condition, whatever the allele it is going to be expressed. But if in any case you need to know that, then you need to have the other allele. So these kind of mutants are used for the complementation analysis. I'll discuss briefly complementation analysis in my next class, and dominant and recessiveness, as I just have mentioned. And they are also helpful in studying of gene regulation. And in this particular case, for making the diploid, no recombination is needed for the gene expression. And what are the limitations? Limited to genes near to the original F integration site. Only genes on the plasmid are transferred and can complicate gene mapping if not identified. So, if it is an imprecise excision, only the genes that are very close to this F integrated F factor will get excised and become the part of this F prime plasmid. Other genes will not be transferred. So just a quick comparison between HFR conjugation versus sexduction. So in the HFR, chromosomal DNA is getting transferred, whereas in abduction, plasmid DNA with chromosomal part is getting transferred. So status of the F minus remains F minus. Remember that hundreds of times this thing is asked, and in case of sexduction, F minus becomes F prime. So recombination required, yes, and in case of sexduction, no recombination is required. There is no diploidy in HFR, but there is a partial diploidy in case of sexduction. So I would end my class today with this activity question, which has been shown at your screen. Read it carefully and answer the question. And in case you feel any difficulty, please feel free to contact me, and you can drop me emails, or in the chat box you have, if you have questions you can write. Dear students, with that I stop my lecture today, and I thank you all for your attention, and I hope you get the concepts better.